Welcome back, folks. We are going to start taking a look at some of the different inheritance patterns that we can see in genetics, how our different genes interact to make us look like us and the world look like it looks. And we're going to start off with a little bit of a how did this all get figured out in the first place type thing, looking at the gentleman who came up with the basics of our genetics as we understand them today. And that man was Gregor Mendel from 1822 to 1884. He was a monk working in his monastery. And one of the big things he did was he was a gardener. He grew pea plants. And these plants he started using for his experimentation pretty early on. It was fortuitous because they actually show some pretty clear patterns. It made it a lot easier for him to figure this out than if he'd have tried to do it with something else. And one of the characteristics of these pea plants he was looking at was like their flower color. They had purple flowers and white flowers. They had wrinkled seeds and smooth seeds and yellow seeds and green seeds, all sorts of different things, different traits that he could very easily quantify and say it's this or it's that, which made his data collection much easier. Now, when he did some of these experiments, some of these cross-pollinations, he took the pollen from one of these flowers and put it on the other and saw what he got. When he did that, all of those plants, when you did a purple with a white, want to guess what it was? All the plants were purple flowers for the kids, okay? So that was interesting. Not a single white one there, not a lighter purple, all purple. All looked like the one parent flower that was purple, okay? Then what he did, something pretty interesting, he took that purple flower, or those purple flowers, those kids of that first cross, and he decided to see what would happen if he mated them together, okay? And when he did that, he got purple flowers and white flowers. As you can see here, he got about 3 purple flowers to every 1 white flower. So out of 4 flowers, 3 were purple, 1 were white. And this happened over and over again. You see, he did it a lot of times. Those are the actual numbers that he got in his experiments. He got 705 purple plants and 224 white plants. A little bit of terminology, just to make it easier for us here. The original parent generation we call P1, the original plants he started with were P1. Their offspring are called filial, okay, like family from the Latin for family. So that we call them F1 or first filial generation. So the first set of kids. And if you mate those kids, then we just go F2, F3, F4. So another way to put it would P1 to be generation 1, F1 generation 2, F2 generation 3. But the standard genetic parlance is P1, F1, F2. All right, so he did this again with seed texture. When he did that, his parent generation was smooth and wrinkled seeds. The F1, all those kids were all smooth. So we start to see a similar pattern again. And when he mated those kids together to get the F2 generation, and he did a lot more of these, he got 5,000, almost 5,500 smooth seeds and 1,850 seeds. But again, if you do the ratio, out of every 4 pea plants, it's about 3 round to 1 wrinkled. Okay, and here's his actual numbers: stem length, pod shape, pod color, all these kind of things. We work our way over. You can see once again, it's about a 5,500, 1,800 again, about a 3 to 1, 3 of one type, 1 of the other, 3 out of 4 or 3/4, 1 over the other. So you saw this over and over and over again. So let's take a look at how this maybe happened. Okay, so he found this pattern, but the next question is, okay, so if I do this, sometimes I get all of one and none of the other, but if I cross them again, I get like 3 to 1 ratio on the 2 phenotypes, right, what they look like. We've talked about that in the vocab already. Didn't have that terminology quite yet. Interesting fact, Mendel figured all this out before we knew about DNA and chromosomes. Remember Watson and Crick, that was the 1950s. They came up with a model of DNA and started to figure out how it worked. He did this all in the mid-1800s, about 100 years earlier. So he didn't know what the internal mechanisms were. He didn't know about genes, didn't know about chromosomes, didn't know about protein expression. But he was still able to come up with a way to predict what would happen in different situations. The heart of genetics for us. How can he explain this pattern and predict what would likely happen? Okay. So let's take a look here. He took his purple and his red. What he came up with was that every plant for each of these traits, like flower color, there were, in the cases he was looking at, 2 versions of those traits, which we now call alleles. So we have a gene for flower color. That we'll use the terminology we use today. Got a gene for flower color, okay? And that gene, in this case, has 2 different alleles. One would produce a purple flower, and one would produce a white flower. Okay. Now, then what he surmised is that each parent, in the process of producing offspring, so basically having sex in this case, the way plants do anyway. So this is why we got this one over here drawn like a sperm, even though plants don't have sperm. We drew this one like an egg, even though plants don't have necessarily what we would think of as eggs. The generic term is gamete, but we're just going to draw sperm and egg because it's very visually evocative and really sinks into your head what these things are. The sperm and the egg, these gametes, the pollen and all that, they only have half or one of those 2 versions of the gene. So over here, this flower could either get that allele for the gene passed on or that allele for the gene passed on. So only one of those 2 alleles winds up in the egg. Same thing for the mother in this case. Only one of those 2 white alleles is passed on in the egg. And that means when we fertilize that egg and they come together, we have a new plant that is back up to the original number of alleles for every gene. So we got 2 alleles for every gene, in this case, a purple allele and a white allele. Okay. And that, if you look back at his numbers, produced all purple plants. So what's going on here? Why is he creating all purple if he's got half the alleles for the color gene are purple and half are white? Wouldn't you expect to be somewhere in the middle? Well, sometimes that happens, but this is where inheritance patterns come in. Sometimes one gene will override another. Sometimes they'll mix. Sometimes they'll both show up. There's a lot of different things going on. That's one of the things we're going to look at in this unit. Okay. So now let's take a look. Let's take 2 of those F1 generations, those kids that were half and half, what we call heterozygous, because hetero means different, right? Somebody who is heterosexual, like somebody of the opposite sex, of the other sex. So this is heterozygous for zygote, one of each. On the left-hand side of the screen, you're seeing a pair of homozygous chromosomes. This one here has, let's say, B genes. Maybe B is for, I don't know, blue eyes. Okay, so this one on the far left has got 2 versions of the B gene, so 2 different alleles, and they both have to be big B's. The one right here has got 2 little b's on it. You know, same version of the gene for eyes here. So we got 2 little b's. These 2 are homozygous. Homo meaning same. Okay, and it can be either homozygous dominant, like the big B, or homozygous recessive, like the little b. On the right-hand side, we have heterozygous. Hetero means different. So here we can see they still have 2 versions of the B gene, but here we got a big B and a little b. And the other one's got a little b, then a big B. Doesn't matter which order they're in, technically, but that is heterozygous. Now, in this case, they're still each going to be able to pass on only half of their genes, or 1 allele for every gene in each particular egg or sperm. So in this case, with these heterozygous ones, one of the sperm is going to have the purple allele in it, the other one's going to have the white. Same thing with the eggs. So what we did here is something called meiosis, where you take these genes are on different chromosomes. You have 2 of every chromosome in your body for a total of 46 chromosomes, in humans anyway. 23 of those you got from your mom, 23 of those you got from your dad, right? And now those chromosomes come in pairs. So you get this first chromosome, and on this chromosome you got from dad, it's going to have hair color and eye color and lactose intolerance all on this chromosome, and the one that matching chromosome for mom is going to have the same thing, hair color, eye color, and lactose intolerance on it. Okay, so you got 2 of those in you. One winds up in one of these sperm, one winds up in the other. Same thing for the eggs. Okay, that's meiosis. That's the production of gametes or making egg and sperm. You got to have the number. So these egg and the sperm in humans only have a total of 23 chromosomes. Now, what's got to happen next is we got to figure, okay, which egg is going to get fertilized by which sperm? You know, there's a lot of random chance, right? Just like every time you have a kid, you know if you're going to have a boy or a girl. It's about 50-50. Well, here, if that's the egg that gets fertilized, and it's fertilized by that sperm is the one that happens to win the race to get into the egg, you can see we got two purples. If that same egg is fertilized, but the other type of sperm gets there, we got a purple and a white. If the white egg is the one that gets fertilized by that purple sperm, and then here we got if the white egg gets ovulated and it gets the white sperm gets here. So there's lots of different combinations of various sperm and egg that can happen. And this is what part of what gives us such a variety. If we do that, that one is purple, right? Because it's got two purples, definitely purple. Just like we noticed before, as long as there was one purple allele in the previous examples, the plant was purple. And the only time we saw a white plant was if we had two white alleles, and so we get that white one. So here we can see that ratio demonstrated very clearly in this thing, which we call a Punnett Square. Oh, I am a bad speller. Is it 1T? I think that's right. That's a Punnett Square for us, guys. And that is how Mendel figured out how to do this. Because you could do all sorts of mental gymnastics, but if you just put in this nice little graph, it's really easy to figure out what are all the different ways that the different eggs that are produced by the mother can come in contact with the different sperm produced by the father. And then we can get some idea of predicting, oh, if we mix, this mother up here with the purple and white allele with this dad with a purple and white allele, what are the chances we're going to have a kid who has got purple flowers or a kid who's got white flowers? Okay, so, and that basically is the principle of segregation or separation. Each individual has two copies of each gene. Those two copies are called alleles. Each parent can only pass on one of those copies to the offspring. So you get one allele for color, for flower color from your dad, one from your mom. When they come together in fertilization, you're back up to a full set, two copies of that gene. Now it's how those two copies interact that then determines what you wind up looking like. Are you a purple flower or are you a white flower? To use the pea flower examples here. That is principle of segregation. We have to segregate or separate the alleles or the genes or the chromosomes, however you want to think of it, during meiosis, the process of making the egg and the sperm. And then during fertilization, we bring them together and get back up to our original two copies of every gene, two alleles for every gene, to make us us. This is the other way you can do it besides Punnett Square to get all the different possible combinations. As you can see, it's kind of messy looking. Punnett squares should not scare you. They're much easier than the alternatives.